more. So the title is Are We Building on the Shoulders of Giants or on a Nanobubble? And so I should explain briefly what this means. I think the nanobubble aspect is quite easy to understand. It's this idea that a bit like the property bubble or the financial bubble. That's, we invest a lot of things into an idea, maybe too much, and at some point, problems emerge, and then suddenly things collapse. I'm not actually really going to talk much about this idea of a bubble. Really, I'm more interested to discuss this idea of the shoulders of giants in this talk. So there is this phrase attributed to Newton that if I've been able to see further, it is by standing on the shoulder of giants. And in fact, the idea is even much older, and this is like from about 1200. And this guy on top on the shoulders is us. We're basically trying to look further, and how do we do that? Is by standing on the shoulders of. So in this context, it's obviously a religious imagery, but it's the same idea that we can learn more about the truth by building on what everybody else has done before. And in our case, it's a scientific record, right? The discussions at conferences and so on. So this is what we are supposed to build on. But are we? And in fact, who in this? Oh yeah, I should have said something else in my introduction. Which is this is going to be interactive, so I need you to answer questions. So obviously, so who knows where this comes from? Remember Sammy Jenkins? No one knows. Come on, no one knows. So okay, well at least you will learn something from this talk. Okay, so this entire talk is an excuse for me to mention this amazing movie, Memento. And since you don't know it, I have to show you a few seconds of it. Oh, the sound is a bit low there.
Yeah.
I shouldn't have to ask.
But even if you get revenge, you're not going to remember it. You're not even going to know that it happened.
My wife deserves vengeance. Doesn't make any difference whether I know about it. Just because there are things I don't remember doesn't make my actions meaningless. The world doesn't just disappear when you close your eyes, does it?
Okay, the world doesn't disappear when you close your eyes. This is actually an amazing movie, and this guy has a very special condition in which he only remembers what happened in the last 3 minutes. This is after a very dramatic accident, and he only remembers what has happened in the last 3 minutes. And this is a big problem in his life, because once he's got a big job to do, and his job is not to discover a new cancer and a medicine, but to have a vengeance on because his wife has been murdered. But the problem is, when you remember only the last 3 minutes of your life, is that it's quite easy for others to manipulate you as well, and so to make you do things that maybe are not the thing. And the scientific record, in the case of this movie, is actually, because he only remembers the last few minutes, so he actually writes things on himself. And obviously, I've lost the microphone. So the scientific record is stuff that he writes on his scale. We don't have to do something so dramatic. But coming back to science, we can't build on something we don't know or don't want to know. Okay, so I'm going to come to you back with some maybe easier questions. So, do nanoparticles cross the blood-brain barrier? Who thinks that the answer is yes to this question? 1, 2, 3, 4, 5, 6, 7, 8. Okay, I don't know. Who thinks that they don't?
Oh, I'm not.
1, 2, 3, 4. Okay, so it's actually quite interesting. I mean, I don't want to comment on the age of people, but actually the answer no is mostly from people who have, let's say, more experience.
But maybe there is a third answer.
Maybe there is a third answer, but really, this is
like the referendum
They
question.
cross
right. Well, but exactly. The third answer is that it's a bit messy. We don't really know. and we should do some more research about it. And that's a really good answer because you can apply for grants to do that. So do nanoparticles cross the blood-brain barrier? There was this paper in 1995 that reported, so you can see it's quite interesting because it's talking about colloidal polymer particles and then nanoparticles in bracket. It's probably one of the first papers that start to use the word nanoparticles in this context. And they said these peptides, which induce analgesia, can go through the blood-brain barrier. It's a quite highly cited paper, it's about more than 300 citations, and I think it's been quite important, even, it's not thousands of citations, but it's been quite important because it triggered a lot of research in this area, and it's still cited today. And this is really a bit of a strange idea because there have been a lot of research for decades about the blood-brain barrier, showing that basically that for things to go through, they have to be really, really small, and this is like from 1974. And in fact, in 1999, there was a paper basically showing that the way these particles, the way that peptides reach inside the brain is in fact because the particle itself induces toxicity and damages the brain barrier. So it's got nothing to do with the fact that it's a nanoparticle. In fact, it's got something to do with the fact that it's toxic. But obviously that second paper is much less cited. And so we don't really know, and we've get to this situation where, I don't know, 20 or 30 years after these events more, we don't really know what's going on. Nanoparticles readily travel throughout the body. Let's forget the blood-brain barrier and talk about other things, yeah? I mean, probably the answers are going to be a little bit the same, kind of, I'm going to, just for the interest of time, not let you actually answer. But the thing interesting about this is that nanoparticle readily travels throughout the body is a literal quote from a review. Very influential review, 2006. It's got 5,000 citations, that review. There is no citations to support this claim. But a little bit later in the paper, there is a citation to that other paper which claims that particles can just basically diffuse through the membrane of cell. And again, this is a completely incredible claim. Because it was challenged very soon after. So the first paper, 2006, 5,000 citations, the next one, about 850 citations. The one that shows this beautiful graph, one of the most beautiful graphs you will ever see in the paper, it shows diffusion of nanoparticles across the lipid membrane at 0 for 4,000 minutes or something. That paper has about 60 citations. Yeah? But it's not only that it was immediately shown to be wrong, it's also that nanoparticles have been used for, again, half a century or more to look at the process of endocytosis. And this is a completely well-known particle, and we've heard it many, many times in the last 2 days. So it is well-known, and there is a lot of things. And yet, you will see these claims again and again and again and again in many, many, many papers. And it's really important because it informs also safety evaluation, attribution of grants, and all sorts of things, investments. Okay. So, last one, the protein corona changes nanoparticle properties. I'm not going to ask you, because, this is actually true, yeah? This is true. It's also completely trivial, and so, nanoparticle protein corona, these are like 3 words, but nanoparticle is like colloids, right? It's been studied for decades before the word nanoparticle started to be used. Protein adsorption on surfaces and on colloids has also been studied for decades before this expression started to be used. And really, even this idea that the protein corona changes nanoparticle properties in terms of drug delivery and so on, it's well studied in the 80s, and I think there were some examples actually in one of the talks about liposomes in the 80s. And so, again, but if you say, we don't really understand interaction of proteins with nanoparticles, you can get grants, and certainly a lot of people got that. So I'm going to switch now a little bit. Yeah, so we can't build on something we don't know or don't want to know. So if that's one aspect of a problem with the scientific record, with the giant shoulders. Yeah, how solid are they? But now, what about things which are maybe wrong. They are in the scientific record, but they're actually wrong, okay? Well, clearly, from this conference, we know that's the case. There are lots of things wrong, in science. I mean, these are just two examples from yesterday, right? But I mean, they said that just in passing, like it was like kind of normal thing, yeah, right? But this is like hundreds, thousands of papers which have been financed by taxpayers' money. And the taxpayer is not going to continue to finance our research if actually all of these papers are meaningless, right? Except ours, of course, but I mean all the others, right? So we have a little problem here as well. So how much of the scientific record is accurate and reproducible? Can we build on the scientific record? The honest answer for the field we're talking about here is that we don't know, because there hasn't been really a reproducibility project looking systematically at nanoparticles interaction with cells, for example, or things like this. So it's not been done. So we can't really know the answer. But based on these quotes and also on other things, there are reasons for concern, and I could spend the next hour talking about the reasons for concern, why there might be a problem with reproducibility, why the incentives are not really pushing us in the right direction, and so on and so forth. And this has been studied and discussed, and is discussed. But I want to focus on just one of the causes for concern, and that is about the mechanisms of self-correction of science. Okay, so we always say science is self-correcting, right? I mean, you agree?
I agree.
You agree? At least one person agrees. Well, I don't. Absolutely not. Absolutely not. For two reasons. First, the first is maybe playing with words a little. You can argue, but science doesn't do anything. We are doing things. Okay? We are scientists. We correct the scientific record, or we don't. Okay? But that you can argue is just playing with words a little bit. But actually, there are many examples of claims which were completely wrong and which were maintained and not corrected for very long times. Tens and tens of papers, years, decades even, before things are actually corrected. Or maybe they are never corrected, they're just slightly forgotten on the side and then nobody works on it, and that's it. So what happens when scientists try to correct the scientific record? So I've tried. So maybe we can learn a little bit from that. I'm going to tell you two short stories. I could spend an hour on each, so I'll give you like 30 seconds on each, because I want to tell you a few more things. But before I say that, also based on discussions I've had during this conference and other things that have happened, this is now entering into the more controversial. This was like the not controversial so far. So it's entering into the more controversial, and the reason it's entering into the more controversial is because it's becoming personal, right? Because we are scientists, but we are human persons, we are people, we talk, we argue, and so I've said to correct the scientific record, we as scientists need to do things. But obviously and that means basically talking critically about other people's work. These people, but they are also our mates, they might evaluate our proposals, they might review our papers and so on. And so we don't really want to do that. It's kind of and also if you work a very long time on something and someone comes and say, actually in your paper you said this and this, but actually this is wrong for this and that reason, you're going to take it personally. Whatever, even if I put it very, very politely, you're going to take it personally. And so we can't avoid these kind of feelings, but if we don't do it, then I don't think it's, we can't actually make this scientific record stronger. And so I would argue that when things are wrong, we have to actually say something about it. So the first story is striping nanoparticles. I'm really not going to describe this, but it's basically a mixture of two ligands on nanoparticles, and they have all sorts of magical properties. I would argue that, I have argued that, in fact, their structures come from a well-known scanning probe microscopy artifact. And it took a very long time to get out, and so the striping nanoparticles, a large number of papers published on these in high-impact factor journals and so on. And at some point, we tried to publish a revisited paper. That took 3 years to publish. When finally it came out, there was a little bit of discussions on social media and on my blog and elsewhere, and then some follow-up papers and a little bit of coverage in the sort of specialist journals. What I've learned from this is if you try to correct the scientific record, it's slow and it's quite depressing at times. I could read the emails I've exchanged with the editor of Small, which eventually published the journal, but it would take another 5 hours, so I won't do that. But scientific journals are quite reluctant to correct the scientific record. That's an understatement. Institutions protect their reputation, not the scientific record. So there would be some investigation maybe, but the point is never to correct the science. Really not. That's not really the idea. Access to original data is key, and I think blogs and social media can play a very important role. And I'll kind of expand on this a little bit. Okay. So the second story is about spherical nucleic acids, and. So in the 2006 there was a science paper, and then in 2007 there was this JAX communication, and so these particles, you've seen them in the cartoon earlier. They're gold particles. They've got DNA on them, and I can see I'm running already over time, so I really need to hurry up. So they're supposed to be able to detect mRNA detection, and the nice thing about this story is that the technology was commercialized. So not only there were papers in Science, Nature, Nanoletters and everything, but also the technology was commercialized. So by a big multinational company, it then became part of Merck. So you could buy these particles, and they were supposed to detect mRNA in cells. Now the problem is very simple. And
our RNA detection reagents use a breakthrough nanoparticle technology, which allows you to detect RNA in living cells, delivering valuable information to your scientific. What if you could detect RNA in living cells? Traditionally, RNA detection has been accomplished through interrogation of lysed or fixed cell populations and involves amplification techniques in order to properly detect signals. Detecting RNA in live cells, where you're examining native RNA species, would provide more physiologically relevant. What if you could detect?
In the interest of time, I'll just explain the problem. So these particles are supposed to come inside cells. The problem is they never explain how they come out of endosomes. mRNA are not in endosomes, so it cannot work. This is the 50-second summary of the controversy. We published the paper, and this guy who was supposed to be developing the project basically told us we were right. Maybe you've had time to read that. Yeah? Okay. So I'll finish in basically 1 minute, if that's okay, with some ideas about how we can do things a little bit better. So that's the first one. Okay, so this is one recommendation, and really there is no excuse for not doing this anymore, because you can't publish negative data. You publish it as a preprint, publish it on a server, whatever you want, but share it on a blog, whatever. Even if doesn't have a DOI, just don't keep this kind of information for yourself. Share it. There is no reason not to do that. And the other thing is, okay, just one more thing about the smart flare thing. Is that actually the product has now been withdrawn. Okay, so it's not on sale anymore because it doesn't work. And then the last thing I wanted to mention is that we need to share critical insights about papers, right? Because what happens otherwise is exactly what's illustrated exactly by the answer to my first question. A few people who are in the know-how, yeah, because they go to more conferences, because they have more experience and so on. They know they can read through the bullshit that is in every single paper, right? So they know that when we say particles can go through biological barriers, this is actually bullshit. It's not true, right? But for the young people who are coming into the field, they have to painfully go through this process of discovering that a lot of the literature is wrong, and that even respected people actually are lying. Let's put it in this way, because that's what you have to do to get grants. So I think we have to share critical insights about papers, and one of the ways to do this is through a website that is called PubPeer. So some of you may know it, others might not. So this is one sort of experiment that I did I took 20 very influential papers, influential in the sense that they were really highly cited, about nanoparticles in cells, and I kind of reviewed them first on Twitter, and then I used this website PubPeer, and I'm just going to show you one example of a, okay, let's just look at this one, for example. And so this is a paper, you know, in Nanoletters 2005. It really doesn't exactly matter. And so I just reviewed it, and in this case, it's a paper which has a lot of problems. I have quite a bit of experience in this field, so maybe what I'm saying could be of use to other people. But what I'm saying is that we should all be doing this, basically. So this is my review, and I just do one last bit of advertising for PubPeer by saying this, is that if you actually install this in your web browser, because journals are not helpful, they don't want really to have commenting platforms that work. So if you install this browser on PubPeer, this plugin in your browser, you can then, when you are on the paper or when you are anywhere that cite that paper, you can actually see that there are comments, and then you can just click on it and see those comments. And with that, I can see my chair is getting a bit exercised, so I will just close and take any questions if there are time for any questions. Thank you very much.
Thank you.